Well, greetings, folks. I was here about 10, 11 years ago. I'm glad to see the tribe is healthy and vibrant, and it's really wonderful to see so many younger people here picking up the torch and carrying it forward. But this talk is something, I hope this talk will forever change your lives for the better. This talk, I have to set the groundwork and the framework of a series of events that have occurred in my life. And these events, when you string them together, it comes to a paradigm-shifting discovery that I truly think is revolutionary. And these ideas are much bigger than the messenger that presents them. I believe in that nature is intelligent. I believe in that we are only witnessing and discovering things that nature has already invented. But we are truly Neanderthals with nuclear weapons, stumbling around this planet, so cockeyed and full of hubris and so attached to our little computer devices that I think it's really important. And this group here represents the body intellect of the earthlings that are connected to nature, that are connected to the soil, that has a much better sense. And so as a scientist, we are really trying to put metrics, intellectual metrics, creating language and words to be able to communicate ideas. But nature doesn't need to have our language. Nature has its own language. And so what I'm going to describe to you today is sort of a lifelong story that I am privileged to be in this body at this time. And I also want to dedicate this talk to my good friends Toby Hemingway and Dahinda. There are two great brothers who have passed on, and I think a lot of us here were greatly influenced and touched by their kindness and their mentorship. So, this is an artist friend of ours made this painting for me, and this really describes towards the end of my talk is the connection between mushrooms and bees. Now, I like this painting a lot because these are actually all taxonomically correct. And being a mycologist, it's really important that we don't have Smurf mushrooms, up on the screen. So I'm going to rock and roll. I will have to go through this fairly quickly because it's important that I get to the end. And I only have an hour and 15 minutes to do an hour and 45-minute talk. So here's a recent article that came out in Science. And unfortunately, over 58% of the ecosystems on this planet now have fallen below the threshold of species diversity that can maintain those ecosystems and the residents they're in, which means external inputs need to be brought in order to sustain the life systems of the populations. We have entered into 6X, the sixth greatest extinction event known in the history of life on this planet. But this extinction event is not caused by an asteroid impact. It's caused by an organism, by us. We're not only the cause of this extinction event, but we're likely to be one of its victims. So it's an all-hands-on-deck moment, folks. And we need to have actionable, ecologically rational, and scalable solutions. This is really important. We can't remain philosophers and idealists. We have to have boots on the ground. We have to be able to put into place remedies and solutions that are sustainable that will have a dramatically positive impact. And investing in biodiversity, I think, is absolutely key to this. So this is a representation. This is the actual pictograph from northern Algeria, then re-represented here, of the bee man. It's a bee shaman figure from 5000 BCE, about 7,000 years ago. And it's very interesting because the artist was clearly trying to communicate that the artist is pretty excited about mushrooms, don't you think? But amazingly, in 4 peer-reviewed articles, the authors dared not suggest what these mushroom-like shapes were. is a representation of mycophobia, the irrational fear of fungi. Even in academia, when you mention mushrooms, lots of people make a bad joke about being in the dark, being fed you know what. And so that sort of hindrance of not scientists being reluctant to talk about something that is verboten in academia was really my opportunity, because I've always been attracted to that which has been forbidden. And so when I was told that mushrooms were scary and dangerous and could hurt my twin brother when I pelted him with puffballs, when I was 5 years old, I said to myself, "Thank you, Mom." But this is unfortunate because it also has permeated into academia. Interestingly, mushrooms were very much used in beers, in fermented honey as well as beer, but the Beer Purity Act in Bavaria in 1516 specifically banned mushrooms. This, I think, is a struggle between monotheism and polytheism. The Germanic tribal people were celebrating in the forests, and monotheism with Christianity came in with the church and said you had to go to a church in order to see God. And I think that is representation of that division in philosophies. But going back, this is 400 years BCE. This is a codolith showing Demeter giving Persephone a mushroom. And upon ingesting the mushroom, Persephone goes into a deep sleep. In Greek mythology, this is the onset of winter. And then upon the spring, she comes from the underworld, from Hades. She comes onto the landscape again, and that is the return of the longer days, sunshine, and the rebirth of plants. So interestingly, the way that Demeter is presenting this mushroom, for those of you who've ingested psilocybin mushrooms, this is the sort of rite of passage and respect. When you present a psilocybin magic mushroom to a person, it's take this. This is special. You know, this is not like a porcini where you put on a table. This is like, this is really special. So 450 BCE, approximately, Hippocrates was using amadou mushrooms for cauterizing wounds, and Dioscorides mentioned agarikon. Now, amadou is actually what this hat is made from. This is a birch polypore. It's a very hard mushroom. And this mushroom, when you put it into water, lye water, it delaminates into a fabric called mycelium. You'll see lots of mycelium. So this mushroom in lye water becomes a fabric, and some ladies in Transylvania are still making these hats. Now, there's only 5 hat makers currently. There used to be 20. It's a thread of knowledge that goes back thousands of years that fortunately has not broken, but how many threads of knowledge from our ancestors have been broken because of disease, war, famine, poverty, accidents, whatever. But this mushroom is known as a fire starter mushroom. It enables you to carry fire for days. And moreover, you can hollow this mushroom inside, put embers of a fire, and then rekindle it. There's no doubt that we all came from Africa. We had 2 migrations in Neanderthals and then Homo sapiens. About 40,000 to 80,000 years ago, there was an amazing enlargement of our prefrontal cortex. This speaks to a different lecture that I'm giving elsewhere on microdosing and the evolution of consciousness using psilocybin mushrooms. But the amadou mushroom allowed not only the portability of fire, but it revolutionized warfare. Because this was the punk that would make flint spark rifles ignite the gunpowder. Moreover, interestingly, this was used by beekeepers to smoke hives. Amadou also is the first mushroom ever to be found to have an antiviral effect against the tobacco mosaic virus. So very interesting. And I think that mushrooms, plants, animals become shamanistically important with indigenous peoples because of a multiplicity of benefits. And indeed, I think this is one example of that. So amadou, because of its flammability, was used for cauterizing wounds. And another mushroom you'll be hearing a lot about is called agarikon, that Dioscorides first described as a treatment against consumption, later thought to be known as tuberculosis. So I'd like to give credit to my teachers. This is Dr. Michael Bugh from the Evergreen State College, Alexander Smith from the University of Michigan, Dr. Daniel Stuntz, University of Washington, and Kit Skase from Post Falls, Idaho. These three individuals adopted me sort of as their mascot when I was about 18 years of age. They've all passed on now, except for Michael Bugh. It's all the more interesting they took me under their wing because these were basically politically conservative individuals, very much concerned about the surge of interest in magic mushrooms. But all the more interesting they took me under their wing because when I was 19, this is what I looked like. Your suspicions are now confirmed. Okay, so the old growth forest is where I spend a lot of time. For 3 years, I set chokers in the woods. I cut down the old growth forest. I'm a strong believer in the school of hard knocks. And I also walk in academia. But going and repaying the old growth forest is something that's really important to me. So on Sundays, this is where my wife and I go to church. But let's look at a cross-section of an ecosystem. You have saprophytic fungi that are breaking down wood. You have endophytic fungi that are growing inside of trees, literally, and plants. Literally hundreds of species of endophytic fungi now have been found to be part of plants as part of their host defensive resistance against pathogens. You have mycorrhizal fungi. Most of you know about that. That expands the rhizome and the rhizosphere enormously by many orders of magnitude, helping the absorption of minerals and nutrients. The parasitic fungi, which are like the wolves, calling the genome and actually helping screen out the weaker individuals and helping to fortify the genome of the survivors. But it is now known that all plants are part fungus. These are giant consortia, giant guilds. And it was thought for a long time that when you do thin sections under the microscope, that when you saw these fine filaments growing in association with plants, that they were infections. But now we know that the majority of these actually are endophytic fungi that are helping the plants survive. So this is important, especially for botanical medicine. You do an extract of a plant. What's the benefit from the fungal endophytes that are in the plant versus the plant themselves? Can you actually have a plant without endophytic fungi present? You can under sterile culture, but you'd have a lot of effort in order to produce a fungus-free clone in vitro. So the way of nature is a plurality of these fungi, consortia, that are working together in concert. Excellent book is Mycorrhizal Symbiosis. This shows you the expansion. The majority of all this is mycelium. And an excellent article came out 2 years ago. It's a very simple experiment, and I'll quickly describe it. There's 5 bean plants. They're in separate pots. They exposed the first bean plant to aphids, and that bean plant upregulated alkaloids that had anti-aphid properties, part of its defense system. The 4 other plants, remote from contact, did not. They repeated the experiments, and they put all the bean plants in the same soil, so the roots would interconnect, and the fungal networks would also interconnect. When they introduced the aphids to the first plant, what happened? All the other 4 plants, remote from contact, upregulated the same alkaloids that were anti-aphid. This showed definitively that there's a communication system underground that is where the community of plants are benefiting from the fungal networks that are alerting them to a pathogen or a parasite on the near event horizon. So this is how quickly the mycelium grows. That was 10 days in a petri dish. And the mycelium is beautifully organized and has many different forms. But many people don't realize that fungi eat rocks. The mycelium consumes rocks, takes the minerals, and then channels them to plants. And so I would see these rocks in the forest, and I thought, that's interesting, there's moisture down there, that the contact. No, that's actually a lens of mycelium that's consuming these rocks. So fungi were the first organisms to come to land over different estimates, 600 million years ago, 1.2 billion years ago. Plants soon followed. Plants and fungi allied together to form lichens. And so in the mycorrhizal fungi soon enabled plants to march across the landscape. So we take wood chips, we add mycelium, lots of things happen, but ultimately we generate soil. Fungi, the grand molecular disassemblers of nature, the soil magicians. And worms specifically chase mycelium. So myceliated habitats in vermiculture, worms specifically choose myceliated substrates over those that are not. I like to show this one because it shows the output from mycelium as it breaks down wood or straw or any other type of plant material. What people are quite surprised and don't know, that 100 kilos of dried sawdust will give you 20 kilos of water. The mycelium inhales oxygen, exhales carbon dioxide, breaks down plant material, combines hydrogen with oxygen, forming H2O. The mycelium produces enormous amounts of water. That's why there's sweat piles, puddles around compost piles. And so in doing so, it hydrates its habitat in advance of contact. So this is a really important article that came out on the unfortunate logging practices that's pushing monoculture and destroying the mycosphere, destroying the resident saprophytic fungi, because you don't have a variegated canopies. You end up with similar aged trees, for instance, and so you don't have the cycling of fungi that you would normally have. What is really interesting to me is that 70% of soils are composed of microbes, but 30% of the soil mass is fungal. So that's an extraordinary amount of fungi that many of you know from no-till how important these fungal networks are. The mycelium, some of my electron micrographs, in response to 4 primary environmental stimuli: introduction of water causes evaporation, drop in temperatures, number 2. As the mycelium comes up to the surface because it's moisture, it exhales carbon dioxide, inhales oxygen, that's number 3. And surprisingly, the 4th one is light. 99% of all mushroom-forming fungi require light. If they don't have light, the mushrooms will not form. Even though they have no chlorophyll, they are extremely phototropic and photosensitive. So upon that, then the primordia begin to form, and they quickly enlarge to a mushroom within a matter of 3 to 4, 5 days. Now, why are mushrooms so feared? Why are they so misconstrued? Why are they so underappreciated? Well, I think for a lot of different reasons. In our viewscape, as we walk through life, we have daily, weekly, monthly, yearly encounters with plants and animals. We have a familiarity factor because we become acquainted with them. But something that is so potent, that can kill you, that can feed you, that can send you on a spiritual journey, and is only up and gone in 4 or 5 days, is very ephemeral, very temporary. And so it's natural for humans to avoid that which they don't understand that is so potent. This is why I think the field of mycology has lagged behind some of the other sciences and not being fully appreciated. I'm fascinated by how mushrooms rot. Here's a mushroom russula in the old growth forest in a meadow. And a few days later, lots of sporulation is occurring, spores are germinating. And then a few days later, the mycelium goes subterranean, goes underground. The mycelium, as it grows underground, has bundles of nuclei, sometimes hundreds of nuclei per cell, streaming through the mycelial networks. This is a movie by my friend Patrick Hickey. And before he made this, we didn't know that these big clusters of nuclei would travel in concert. Now, if you look very closely, they don't all go in the same direction. A few of them go in the other direction. So in a swath of mycelium, the breadth of my hands, my arms here, you can have hundreds of millions of end branchings. Now, the internal part of the mycelium is only 2 nuclei per cell, typically. But at the tips, there can be hundreds. Think of them as little scientists. And at the tips, they're encountering new potential food sources, new toxins, new food, new plants, whatever. If there is a recombination of genetic material that expresses a new enzyme that successfully captures that food and breaks it down, what happens? The mother mycelium becomes educated. So these are not only externalized lungs, externalized digestive membranes, but they also have the ability to learn at exceptional paces. So I live in Washington State, in the southern region of the Puget Sound. Here's the Columbia River. We're going to do a fly down here to the largest organism in the world, is a mycelial mat. 1,665 football fields, 2,200 acres in size. And think of it, and it's 1 cell wall thick. Right on the other side of that cell wall is hundreds of millions of microbes, many of which want to consume the mycelium. How is it that a membrane, one cell wall thick, in direct contact with hundreds of millions of other microbes per gram, can achieve the largest mass of any organism in the world. It's because of epigenesis and the fact that these are in constant biomolecular communication with this ecosystem, learning strategies to be able to survive, and also predetermining the ecosystems that give rise to the plants, that give rise to the canopies, that create the debris fields that feed the mycelium. These are mothering membranes whose interest is in the long-term survival of the ecosystems, not for its own individual survival. Of course, that comes with it. But in order to have a plurality and biodiversity in the ecosystem is absolutely critical for these fungal networks to be able to thrive. Whew, that was a tongue. So in this case, it's the honey mushroom, Armillaria astoi. And I'd like to go back on this because I hired an airplane out of Boeing Field. I flew there twice, and we couldn't find it. And we went back, and in the scientific literature, there's latitude and longitude readings. So we went back, we were right on top of it, but we weren't high enough. So we flew back again, and I spiraled up in this airplane. We got up to about 15,000 feet. And I'm in a little tiny canvas-covered airplane, literally. It's a bush pilot from Alaska. And I said to the pilot, I think I'm going to faint. We just came from sea level to 15,000 feet in an hour. And he goes, me too. I said, okay, I'm going to get a photograph first, but... So this is the best photograph of the largest organism in the world. I beat National Geographic on this one. Anyhow, so the mycelial network, is designed to resist from catastrophia. There's no cell place that you can break the mycelium that'll harm it. It's one of the few organisms in nature where you disturb it, it grows more vigorously. In fact, this is a technique by mushroom growers to scratch their beds to disturb the mycelium because you get another flush of mushrooms. So it's we separated from fungi 650 million years ago. There's a new super kingdom called Opisthokonta that joins Animalia and fungi together because we exhale carbon dioxide, we inhale oxygen. Many of our best antibiotics against bacteria come from fungi, but very few good antifungal antibiotics are out there because they tend to harm us because of our closer evolutionary relationship. So the mycelium also then can be parasitized by bacteria, but a minor population of bacteria out there are actually mutualistic, that the mycelium preselects in order to fortify its immunity against bacterial pathogens, using the bacteria's defense mechanism against other bacteria. So the mycelium sets up guilds of microbes, microbiomes. And some of you were in my talk yesterday. I showed you next-gen sequencing, where there's 1,000-fold difference in the genera of bacterial populations between two different mushroom species grown on the very same material. So we do know they select out microbiomes that are favorable to the plant communities that give rise to the ecosystem that can feed the fungi. So the mycelium sweats these little droplets, lots of water, as I mentioned, but also in them is antibacterial, antiviral, messenger compounds, the whole slew of extremely interesting constituents. There's over 240,000 novel molecules that comprise a mushroom, many of which are not found elsewhere in nature. The mycelium, when it grows, is tenacious. It prevents erosion. This rhizomorph here of mycelium, I cut off a centimeter, weighed it, and I weighed this. There was a 30,000 to 1 difference in the mass, which means 1 centimeter held 30,000 times this mass. When I could have made this 1 millimeter, that would have been 300,000. So myceliated substrates, as we know from no-till, prevents erosion. Those fungal networks, tend to retain the soil and prevent loss of nutrients. The quickly diverging rhizomorphic mycelium is, for me, as a cultivator, is what I relish to see, these rapidly forking rhizomorphs. This is where epigenesis is occurring. This is where polynucleation is occurring. The vigor of growth is just extraordinary. So my wife Dusty and I spent a lot of time in the old growth forest. And in the old growth forest has become a treasure trove of our genomic library. We have about 700 species and strains in our cultural library to date. And we march these things out. Some of them we preserve just for posterity. There's not enough hours in the day to do what we need to do to explore the beneficial ecological and medicinal properties of these fungi. But I am really fascinated with decomposition. This is my manly man photograph. When I was 19, 20 years old, I suffered from testosterone poisoning. A lot of you young men, and for that women also, you do dangerous things. So the most dangerous job in the world was being a logger setting chokers. So I go, well, that's for me. So I did that, and then 3 guys in my crew got killed. And they wanted to make me the hooker. That means the top dog on the crew. And I decided to go back to college. So one mushroom resident in the old-growth forest is called agarikon. It is Fomitopsis officinalis. It's the longest living mushroom in the world. It's a perennial polypore. The amadou grow for 3 or 4 years. This grows for up to 100. These are annual growth rings. I was very interested, and have been, in agarikon. I wrote several articles. And then National Geographic, gave me a Green Innovator Award. And they're fascinated by my interest in this rare mushroom called agarikon. Now, my dear professor, Michael Bugh, who I showed a photograph of, he goes out in the old-growth forest 12, 20 times a year. He found his first agarikon last year. That's how rare it is. And he's an expert. So finding agarikon is not easy. When you do find it in a valley, oftentimes there are satellite colonies and other trees, as one would expect. So National Geographic wanted to do a story on me on a search for agarikon, so they could send a photojournalist out. And we decided, well, why don't we go up the inland passage of Desolation Sound in British Columbia. And they said, well, we don't want you to take us to a site where there's already an agarikon. We want to find a new one. I don't know. Well, National Geographic, I'm like, great, they're coming out. Oh, bad news, they want to find a new agarikon. And they said, well, how likely will we find it? And I go, oh, 50-50, maybe. But reason for that is we got a motor sailor, in a Zodiac, went up the Desolation Sound, and these things look like giant beehives, and where bald eagles are in trees, oftentimes you find agarikon. So you look for a living snag, bald eagles, and so I took 10 of my friends, high-powered binoculars, and we scanned the horizon. Now, we left at like 7:30 in the morning, and by 11:30 at night, we're going up, in that morning, 4 hours later, we're going up Desolation Sound, and we have retina burn. Because, look at a tree there, nothing there, tree there, nothing there, tree there, nothing there. You do that hundreds of times, and you're like, dizzy. And so the photojournalist was getting pretty impatient. Paul, you promised me. And I said 50-50, dude, So we're going along, and so our skipper said, this is not doing too well. It's lunchtime. Let's go over to our First Nations site where there's some interesting pictographs. We don't know what they mean, but let's go over there and have lunch. Now, this is a 30-foot overhang. You can bring canoes, or a boat right up into this flat area. It's, used to run red with salmon. The perfect place to get out of the weather and also have access to the water and fish. So we're motoring over there, and then Jimmy, one of our colleagues, goes, there's one. And sure enough, we found one. This is amazing because this agarikon was attached to an upper limb. It fell, it hit this limb, it teeter-tottered, then it regrew its mycelium back into the tree, and then it grew 2 legs. So it was a really extraordinary agarikon. We got really excited about this, going, wow, this is great. So, the photojournalist was really excited. So we're there, and there's an interesting legend with the Haida people and other first Northwest coastal first peoples about the origination myth of women. And with the Haida culture in particular, this is Raven, and she's traveling on a canoe in the Sea of Eternity. And no one can help Raven succeed in finding her genitalia. She is sexless at this point, except one person in the back of the canoe can help her, and is known as Fungus Man. And it's either agarikon or the artist conch. There's 2 different competing theories for this, but these are agarikons that are carved and placed on as grave guardians upon shamans' graves. They help them go into the afterlife. So agarikon was not only revered by Dioscorides in Greek culture, but with the Haida and the first peoples will also realize that it was a very important medicinal ally, and spiritually so as well. So Gu Zhao is the president of the Haida people. I felt it was my mission to bring back the knowledge of agarikon back to the Haida. There was 2 smallpox pandemics. Some people call it the first bioterrorist acts of the United States government against indigenous peoples, where they purposely gave smallpox-infected blankets, and they didn't even have to fight these warriors. They could walk in the village the next day, in the next year, and they would be dying of smallpox. So Gujal, and this is the artist conch, is the other one that may have been this fungus creature in the back of the canoe, because the artist conch you can draw a map on, and it would make sense that, well, you're in a canoe, you need to find your genitalia, you need a map to it, so why not draw it on the artist conch? So we don't really know whether it's a Garacon or the artist conch, to be fair here, but there's 2 competing myths or candidates for supporting the origination myth of women in the Haida culture. So Dr. Scott Franzblau, supported by the Gates Foundation, the director of Tuberculosis Research Institute, University of Chicago, read one of my articles, got real excited, and said, Paul, I want to come with you. So this is actually one of the pictographs at that site. And there's 2 wide-eyed, and it's very similar. You know, it's made with salmon eggs, by the way, and painted, but it's faded a lot. So Scott was real excited and said, you know, I read about Dioscorides and agarikon being used to treatment against consumption. Paul, we think consumption was tuberculosis. I'm a tuberculosis expert. I'm the head of this tuberculosis research laboratory. I want to travel with you. So Scott and I teamed up. Now, this is classic pharmaceutical discovery with natural products. You take a plant, you take a mushroom, and it's called bio-guided fractionation. And basically, you have a water, the most polar of solvents. You use water, and then you use the least polar solvent, like hexane. And hexanes will pull out lipids, and water will pull out proteins and polysaccharides. Well, you have 2 different solvent extractions, and now you test both of them. Which one showed more activity against tuberculosis? It's a decision tree. Oh, this one over here did. And then you fractionate it again. And then you go, okay, which branch of that fractionation increased potency against tuberculosis? That's a classic way that drugs have been discovered from natural products. So we did this over 4 or 5 years, and sure enough, Dr. Scott Franzblau, Guido Pauli, and myself and other colleagues, we found 2 novel anti-tubercular compounds called chlorinated coumarins, highly active against XDR-TP, multi-drug resistant strains of tuberculosis. So we published in the Journal of Natural Products. It was a classic pharmaceutical discovery path. Got very excited about it, but we, in a sense, confirmed what our ancestors knew, that agarikon seemed to help against tuberculosis. Now, tuberculosis was not resident with the Haida people, North Coast Indians, nor with flu viruses, nor with smallpox. So Gujal's people, when he said, Paul, if we knew about agarikon, we would have used it, but you have to realize, in 40 years, we had 4 pandemics that killed 75 to 95% of the elders and the people in his tribe. So the thread of knowledge was cut, from their ancestors. So, okay, so this is great. We found 2 active molecules, active against tuberculosis from agarikon, supports the Haida use and the use by Dioscorides. And we're at this site, and I'm such an idiot, because I see this rock, but I'm there for hours, okay? And I look at, this rock is really interesting. Whoa. There's that rock. There's agarikon, there's a rock, there's agarikon, there's a rock, there's agarikon, there's a rock. This is how my brain works, right? I'm like, oh my God, we found a rock that looks like, it's shaped like agarikon. Now, how likely is it we'd find agarikon? I don't know, 1 out of 100. How likely is it we'd find agarikon at a First Peoples site that seemed to be really important? Well, I don't know, 1 out of 10,000, 1 out of 100,000. How likely is we to find it where there's pictographs that look really reminiscent, of the Haida origination of the myth? I don't know, 1 in a million. How likely is it we'd find it where there's a rock that looked like it's been carved like an agarikon? I don't know, 1 in 10 million. How likely is it we'd find it on my birthday? And this is when the photojournalist started shaking and looking at me and turned to one of my friends and said, does this happen to Paul often? And my friends looked at him deadpanned and said, yes. And this is something that, as a scientist, and I believe there's a convergence. I'm not religious, but I'm spiritual. There's a convergence of science and spirituality. I believe if you walk with respect for elders, respect for indigenous peoples, respect for the ecosystem, You walk in faith and with good intention, nature and science will reward you. I strongly believe that. So agarikon, then it's hard times to find. You know, this is my friend Scott Baker. He's an arborist and tree climber going up a 700-year-old Douglas fir tree on Cortez Island. This is one of the oldest agarikon we've ever seen, at the time the oldest one. And this is the new growth. This one's about 70 to 80 years of age. It does not rot the branch that supports it, very interestingly. And so we went up and we were able to get cultures of it. Now here's the ghost form with Scott, and Mel has a living form. And so there's a metamorphosis from this form to this form. And the ghost form is what was considered to be extraordinarily important. It's dead. It takes 4 or 5 years or longer laying on the ground, where it metamorphosizes biochemically into this quinine form. It's known as the quinine fungus. It doesn't have any quinine, but it's extremely bitter. So very interesting. So this is the largest one we've ever seen. The NorCal kind of guy, can you tell? Found east of Sacramento on a larch tree, old growth larch tree. And they were going to cut down the larch trees, and this person called up because he'd seen my talks on agarikon, said, Paul, I found a huge one. You know, should I pick it? I go, oh, don't pick it. Let's GPS it. Let's leave it in the forest. We don't want to pick these things. And he goes, well, they're going to cut down the forest. And I go, oh, in that case, pick it. So the loggers are going to come through. They're going to destroy it. So, you know, sure. And so he sent us, I'll send you a small chunk. He sent us a chunk about 2 pounds. So this one's about 100 years of age. We now have 77 strains of agarikon in our cultural library. By far the largest cultural library in the world, and different ecosystems. But we typically just GPS them. We leave them resident. We take a stainless steel tube, a small amount of tissue is taken out, and then we get it in the culture. And so my culture library is probably my greatest personal material asset. If my family, my employees are taken care of, and there's an earthquake or a fire, it's my culture library. Everything else can burn. Everything else can be destroyed. My genomic library cannot be replaced very easily, except for many lifetimes, and many of these ecosystems have already been decimated. So we've lost the genomic library of agarikon within those ecosystems. So I wrote an article, and 2 articles in Herbalgram in June of 2001. And I did a synopsis by doing literature review of all the articles ever published on the antiviral properties of mushrooms and all the antibacterial properties of mushrooms. 2 consecutive articles, a whopping 1 page long. That's all there was in the scientific literature. I was amazed. Penicillium molds, hundreds and hundreds of articles. And this is all the articles that were ever published on the antiviral and antibacterial properties of mushrooms. And half this page is photographs, right? So I was like, wow, that's bizarre, because mushrooms like to resist rot. They want to sporulate. They're in contact with microbes. How weird that scientists would avoid mushrooms for exploration of novel antimicrobials. Well, that was in June of 2001, and then 9/11 occurred. And a researcher, medical researcher, physician, saw my articles and said, Paul, you should really get involved in the BioShield Biodefense program. I'm going to get you invited. So I was invited. And we started preparing specimens, mushrooms boiled in hot water, mycelial extracts prepared in different ways, water and ethanol. And lo and behold, I submitted, there's so many stories I could tell you. I'll tell you one real quick. I got these research results that came in, Federal Express, from the BioShield, from the Department of Defense. And a federal package. I'm flipping through them, no activity. This is against smallpox, no activity, no activity, no activity. I get to sample 77. And it said high activity. Sample 79, high activity. Sample 85, high activity. I got really excited. I didn't tell them what the specimens were. They were coded with the laboratory codes. I looked them up, and all those 3 hits were agarikon. Wow, I got real excited, and I was given a colonel physician who was my point of contact, sole point of contact with the BioShield program. So I call him up and say, this is great, we have great results, I can't believe it, 77, 78, 85, hugely active against smallpox. And he goes, what results? I go, well, Federal Express just delivered them to me. He goes, you're not supposed to get those. I'm supposed to get those. I said, no problem. I'll photocopy and send them to you. So this went on for a while. We had extraordinary results. Then there is a vetted press release from the Department of Defense. It says we have the best results of more than 200,000 specimens submitted. It's actually over 2 million. You can Google my name, Stamets and smallpox, in National Public Radio. You'll hear an interview with myself, the Deputy Director of the BioShield Biodefense Program, and the Deputy Director of the FDA. And I was then blurted out, this means, at my TED Talk, this means that we should save the old growth forest as a matter of national defense. And, whoa. I then, at that moment, learned the significance of sound bites, And the Department of Defense, they love this, So they had calls coming to me personally from other researchers saying, we are so excited about this. And sure enough, so we had very good results against pox viruses, orthopoxes, vaccinia, variola, which is smallpox. And those of you after 1962, I believe, are no longer vaccinated. So many of you before 1962, after the age of 5 years, you have a smallpox vaccination on your arm. The majority of you don't. You're what we call immunologically naive. If there's a smallpox pandemic, you are extremely susceptible to dying. But there's good news. So we started working with the University of Mississippi, the NCNPR, National Center for Natural Products Research. We did the same thing, bioguided fractionation. 2 solvents, we split. We did this 5 or 6 years. We found 2 novel anti-smallpox molecules inside of agarikon, more potent than cidofovir, which is the preeminent antiviral drug control. So agarikon is active not only against bacteria, tuberculosis, it's active against viruses, including smallpox. So it's a consortium of many active constituents. This is a real argument for natural medicine. So we identified these new molecules. It took hundreds of thousands of dollars. So with both of these experiments, hundreds of thousands of dollars were expended in order to find these novel molecules resident and naturally produced within these fungi. Well, very quickly, the BioShield program became very concerned about weaponizable flu viruses. And so sure enough, they said, Paul, listen, we have your samples. We would love to test against flu viruses. Is it okay with you? I said, sure, go ahead. And then we hit another big home run. But to give you an idea, 2.5% of the population of the world died in 1918 from H1N1. It's a pig flu. The poultry flu virus, H7N9, recently came through a few years ago. There's a H5N1 has a 60% mortality for those people who get infected. H5N1 is in about 10 locations right now in Asia, causing quarantines, all over Asia for preventing, because if it combines with pig flu, H1N1 and H5N1 combined, then you have a highly contagious flu virus that will kill the majority of people that get it. And this last flu season, as many of you know, was pretty bad. Just imagine 60% of those people that had flu would die. So then the poultry virus swept Minnesota and Iowa, H7N9, and a person who has a very large poultry business, I helped out his family with a personal medical issue, and he goes, Paul, listen, we're euthanizing millions and millions of chickens and turkeys. It says, can you send us some of this agarikon? So we sent 50 kilos of agarikon to an organic chicken farmer in Iowa, and they consumed it in their feed. It's just mycelium grown on rice. And those 20,000 chickens never got the virus. It became an oasis of immunity. Now, there's millions and millions and millions of chickens and turkeys were euthanized. And so we had sort of our first little quasi-clinical study, with chickens. So BioShield is 4 different agencies that were involved, including NIH, virology. And Dr. Earl Kern is a virologist, and I'm going to give you a metric here. It's a selectivity index metric. Anything of 2 is over is active. 10 or active means you've got something really potent. That's why many of the antiviral medicines that people take have selectivity indexes of 10 or more.
So I just got this data last week, and it's so curious. They tested 2,392 samples from me. I had no idea it was that many. I just kept on sending them in. So I never thought I would speak these words, but I want to personally thank Dick Cheney and George W. Bush for funding this research. Now, I say that a little flippantly, but actually it has a deeper meaning and consequence that I want to lead you to. So I am indebted to the U.S. Defense Department and NIH Virology and the BioShield Biodefense Program, which continued until around 2009. And then one of the researchers, Craig Day, was so excited about our results that he continued the antiviral studies unbeknownst to us until last week, when he told us, you know, Paul, because all he had is the alphanumeric numbers. He goes, the samples were so potent. I was just curious. I wanted to do more tests with them. So we are continuing our relationship now with the Utah State Institute of Antiviral Research, because we have some other extraordinary results against other viruses. So in summary, 2,392 samples, these are the best results. So that's a pretty big data set. So the ribavirin is anything over 10 is highly active, 32, 170, yeah, that's positive drug control. And our extracts were in 35% ethanol. This is in vitro human cell assays. You can't put 35% ethanol in contact with living human cells, it'll kill them. So they diluted them 10 to 1, 3.5, 10 to 1 again, 100 to 1 dilution. So these are our results with 100 to 1 dilution of a natural mycelial extract in water and ethanol compared to a pure pharmaceutical drug control. Off the charts. 10 times more, oftentimes, than the positive drug control active against flu viruses, including agarikon. Agarikon is active not only against smallpox, a DNA virus, but also against flu viruses, which are RNA viruses. From a virological point of view, this is kind of unprecedented, but it's because I think there's many other active molecules resident within these natural wild mushrooms. So we were able to grow them up, and then a combination of 3 of them, the selectivity index is 30 times more potent than ribavirin. So this was really exciting for us to get these research results, and then it began to dawn on me that I'm entering into a field of discovery much broader than anything historically ever anticipated, and especially by me. So the first patent issued in 2014. 2014, this patent was submitted in 2004. It meandered and got stopped in the patent office for 10 years. After 3 or 4 years, I got a hold of my patent lawyer. I said, where is my friggin' patent? You file a patent in 2 years, or a year is up on the patent application homepage. The Department of Defense hijacked my patent because of national security. Wow. It took it out of the patent office. We had to do an intergovernmental agency trace and pulled back the patent out of national security. They had to jury it and didn't think it was a big deal. So we got it back, and a universality of opinion of 10 patent examiners approved the patent. Well, that was great, but then the Russians came out from the Vector Institute, and they discovered the same thing I discovered, but eight years later. Well, actually, that's good news, because the vector is like the Fort Detrick of Russia, where they have smallpox. And then another group of scientists also came out and affirmed what I had discovered 8 years later. So actually, that's good. That means other scientists are also validating what I discovered. Okay, so now we're going to get into a little bit more of the science here. As it turns out, there was a shortage of Tamiflu. Many of you know about that. Rumsfeld has an equity interest, in the Tamiflu licensing, and huge amounts of Tamiflu were taken off the market. It comes from the star anise, which is grown primarily in Afghanistan and the Middle East, and it's harvested once a year. You can imagine the supply chain issues. Tamiflu's precursor is a star anise. As it turns out, oyster mushroom mycelium, when exposed to light, can produce the Tamiflu precursor 24/7 in vitro. Light stimulates an antiviral substance coming from mycelium. This was like, whoa, interesting. So the reishi mushroom, we grow lots of that. My friend Dr. Andrew Weil. Reishi has now been sequenced, and the interesting news about this is over 25% more genetic gene expressions in the mycelium than in the mushrooms. The mushrooms are at the end of the life cycle. To get there was a struggle. over years, months, and then the mushrooms come up for a few days, disappear. So it's logical that the mycelium has a lot more upregulation needs to defend itself against pathogens than the end of the life cycle. So we explored mushroom extracts versus mycelial extracts. Hands down, the mycelial extracts turn out to be more potent. Okay, all this now leads up to the following story. I don't know how to tell this story except by calling them mycofactors. So all of that, believe it or not, was mycofactor 1. I'm going to take you through a mycofactorial equation that ends with an epiphany that I think is truly paradigm shifting. So just clear your receptor sites now. All of that was just setting the stage of antibacterial, antiviral properties from these mushrooms. So here we go. I'm growing the garden giant mushroom. Several of you were in my workshop yesterday. It grows on the garden and on the ground, grows in wood chips. I had 2 beehives. I go out to my raised beds where I have the garden giant, and I look closely and I can't believe it. But there's bees going from my beehives to my garden giant patch, all sorts of flowering plants. There's a convoy of bees for 40 days in the summertime, from dawn to dusk. And they went to my garden giant patch and they moved the wood chips and they were sipping on the mycelium, those little droplets. And I got really close to my camera and I took these photographs. It took me hours and hours in a 120-degree attic going through Kodachrome 64 slides to find these 2 slides. I finally found them. And so I published this in Harrowsmith Magazine in 1988 and one of my books in 1999, and thereupon forgot it, okay? That was mycofactor number 2. So these are experiences in my life. When you string these together, it catalyzes towards a solution. So a beekeeper in Ontario wrote me, said, maybe that's why bees go to sawdust piles in the summertime. Nobody else mentioned it. I forgot about it. I spent a lot of time in the old growth forest, and I'm really fascinated by bears. Bears scratch trees, and when the bears scratch trees, resin comes out, and bees are attracted to the tree resins for propolis to patch their hives. Well, so that's interesting. I had noted that before. And I'm walking the South Fork of the Hoh River with my wife, Dusty, and I like to orienteer. My employees call it the Stamets death march. It's not really true. But I don't call it getting lost. I call it being temporarily disoriented, So we're temporarily disoriented, bushwhacking in the old growth forest beyond the end of the trails. And we go around a corner and we see this bear strike. This is like a bear going, boom, one of the best bear strikes I've ever seen. So I thought, wow, this is interesting. This is kind of a teaching moment. So I said, Dusty, this is really interesting because where we live in Kimilchi Point, Washington, they have killed hundreds and hundreds of bears. The timber industry was hired by the Forest Service to kill bears because they cause a polypore mushroom infection of the red-belted polypore. So humans are so good doing exactly opposite their best interests. Now we know that bears bring salmon carcasses and sea salts and minerals from the ocean that helps the trees grow. But they killed all the bears in our area. But I thought, that's interesting. Let's come back to this bear strike in 2 years and let's see what's growing there. Well, 2 years later, it took us about 6 to 8 hours to find this. And there is the bear strike. There's a photograph that I had before. Bear scratches are entry runes for polypore mushrooms. And sure enough, they had that right. The red-belted polypore, a sister species to agaricon, was growing out of the bear scratch. So bear scratches do cause polypore mushrooms to grow. I went, wow. Too bad they killed the bears, but academically, I could see where they were coming from. They wanted to protect the timber board feet of lumber by preventing the trees from getting a disease. Okay, mycofactor number 3. Okay, all right, bear scratches provide entry wounds for polypore mushrooms. So my friend Louis Schwartzberg is a Nat Geo Walt Disney filmmaker. Say, Paul, the bees are really in trouble. What can you do to help the bees overcome colony collapse disorder? And I said, that's really weird because it rekindled this memory of me having my beehives going after my garden giant mycelium. And I said, Louis, let me think about that. So President Obama came out with a presidential memorandum. Bees are dying from a confluence of stressors: bee nutrition, forage lands, parasites, viruses, pesticides, neonicotinoids, glyphosates interfere with the microbiome of the bees. And so it's a perfect storm of stressors. The bees are the canary in the coal mine, so to speak. Apis mellifera is a European honeybee. It was brought over in the 1700s, again in the 1800s. And 80% of our pollination benefits for farmers come from wild bees. 20% comes from cultivated bees, Apis mellifera. So the use of neonicotinoid and insecticides now shows a reduction in sperm quality, viability, reduces drone lifespan. Here is, thanks to Whole Foods, here's your grocery selection in the dairy department. With the benefit of bees, it would be without bees. 30% of your food is directly dependent upon bee pollination. 60% is indirectly dependent. So it's a worldwide threat to global food supply. This is one of the biggest problems that we face today. The entomologists I'm working with have predicted that within 10 years, some say 5, commercial pollination is going to be over for using bees for commercial pollination. So here is this is going to be difficult. This is a 3-minute movie that showed in hundreds of PBS stations. So I might have to go 5 minutes over.
Wow, very nice. These girls are fantastic. I lift that lid up and those girls are solid across there and they're making honey and they're making babies.
Eric Olson owns more colonies than any beekeeper in Washington State.
Hot dog. There's nothing greater than to open a beehive and see them doing well.
They're doing well today.
Look at that. I'm really tickled with this.
But just months ago, he opened his hives and discovered nearly half his bees were dead.
I spent 20 years as a pilot in the Air Force and my share of combat situations, and I never was as low as I was when all those bees were dead. That's the lowest time of my life.
It turns out this may be the new normal. The U.S. Department of Agriculture says that nearly half of colonies across the country died in the 2014 season. Big losses have been happening for years, and scientists haven't pinpointed what's causing them. They say more than 60 factors may play a role in collapsing colonies. Factors like pesticides, malnutrition, and loss of habitat.
If we don't find some answer, I am really concerned about whether these little girls will survive.
But one unlikely solution may be growing close by, in the forests of western Washington.
Oh, there's another one.
Enter Paul Stamets. He's a pioneer in the study of mushrooms.
This is a beautiful specimen. The white margin here means it's growing really well. It's what I call happy mushrooms. Makes me a happy person too.
I'm involved in the study of fungi ever since a very young age. My initial interest was magic mushrooms, and then I got into edible mushrooms and medicinal mushrooms, and my mother was much happier.
Stamets scours the forests for rare types of fungi.
I use mycology and the use of fungi to help clean up the environment, improve the immune system of animals, and I began to think. We've gone to the moon, we've gone to Mars, and we don't know the way of the bee.
All right.
You know, I bet you I can do something to help the bees.
Stamets recently discovered a mushroom that might be able to take on one of the honeybee's worst enemies.
And that's called the varroa mite, with the name Varroa destructor.
Varroa mites began wreaking havoc on U.S. beehives in 1996.
We lost about half the colonies east of the Mississippi over that winter.
Steve Shepard is an entomologist at Washington State University. He's spent decades trying to understand how varroa mites cripple honeybees. He says they invade hives and attach themselves to infant bees.
I always think of it as having something about the size of a pancake feeding on you.
They live off bee blood and transmit a slew of viruses to their hosts. Some sickly bees lose the ability to fly, and gather food for the hive. Many end up dying prematurely.
They'll kill the colony within a couple of years unless beekeepers intervene.
That's why Shepard decided to try a new approach.
Something doesn't look quite right with that.
Yeah, it'll never fly.
He teamed up with Paul Stamets. Stamets told him about a type of fungus that's highly attractive and highly lethal to termites. Shepard wondered what this termite-killing mushroom extract would do to the varroa mite.
So we should do something with this hive, huh?
Ready?
He recently started testing the product on bees in his lab.
So we take bees from colonies with high mite levels. We set up numerous cages, some with fungus.
They're finding that the product is killing mites without harming bees.
It's certainly encouraging so far.
And that's not all that mushrooms can do for bees.
Bees have immune systems, just like we do. And these mushrooms, they're like miniature pharmaceutical factories.
Their initial results show that certain forest mushrooms can reduce viruses in bees and help them live longer.
I think I've discovered now that the fungi that are rotting the logs are absolutely critical for the immunological health of the bees. This is a really interesting potential breakthrough in understanding how nature works and how we co-evolve with fungi.
Shepard and Stamets plan to expand both experiments by partnering with commercial beekeepers. Eric Olson was the first to sign up.
Yeah, I don't have too much hair left. I have pulled my hair out. We just can't seem to get a control on the varroa mite. We've got our fingers crossed.
The future of Western honeybee colonies and the billions of dollars of crops they pollinate may depend on it.
Well, thank you, Earthfix and PBS. This aired on hundreds of the stations last year. So I have 15 minutes to go through the rest of this. So this is one of the fungi we're using that goes after the mites. And we have a simplicity of a delivery system. We just put the petri dishes directly in the beehive. The bees then spread the fungus, and then the fungus infects the mites. So we have this experiment ongoing. And now we have also been doing this indoors as well as outdoors. So that is now mycofactor number 4. So, okay, Metarhizium, this fungus infects varroa mites. Varroa mites are like dirty syringes. They're injecting all sorts of viruses inside the bees. So at 7% infestation of mites, the beehives are considered terminal. 7%. And that means the beehives will not survive typically over winter. Then this article came out. And I was like fascinated. I'm not a chemist, but I'm really interested in the decomposition of logs by fungi. And this article came out, and they found that the bees, the colonies collapsed. Some of you may not know this. In 2 days, they're gone. It's not like there's hundreds of bees at the beehive. They're gone. There's just a few bees. And so they go and they disappear. And then they analyze the hives that have been abandoned, hundreds of pounds of honey. The group of scientists found, wow, there was no p-coumaric acid inside the honey. Now, I knew what p-coumaric acid was, but I was really surprised to find that p-coumaric acid controls the cytochrome P450 pathway. That is your detoxification pathway. All animals use it. It's how we break down things in our liver, toxins. And without p-coumaric acid, the bees could not detoxify neonicotinoids, glyphosates, herbicides, et cetera. And so I went, wow, well, p-coumaric acid, I've seen that. I looked into the literature, and sure enough, as mycelium breaks down wood, it produces p-coumaric acid. So it dawned on me, oh my gosh. So I'm laying in bed, and I have this waking dreams, and I like to stay in bed and just before all my neurons start firing and just think about the milieu of ideas. And I had this epiphany. I go, oh my God, I think I know how to save the bees. And so so p-coumaric acid comes from mycelium. It's in these droplets. It activates the detoxification pathway. Deforestation and the lack of decomposing wood it eliminates the p-cumaric acid, which is also in some cereal grains, but primarily it's in wood as fungi breaks down wood. So it turns out that glyphosates suppress p54 enzymes in the gut microbiome and causes disease of bees. So you want an argument against GMOs? This is it. Glyphosates interfere with the microbiome of animals. Okay, so the mycelium is coming up to the surface because of moisture. It's exposed to light. Light then stops the mycelium from breaking down wood and triggers it in a mushroom formation. So when you have blue light in particular, it causes primordia to form, and we know that blue light causes produces the precursor for Tamiflu. So I'm going, oh my gosh, this is starting to be really interesting. That was mycofactor number 5. P-cumaric acid activates detoxification pathways. So we started making myco honey and feeding it to the bees. This is a form of honey that there's no honey involved. It's just mycelium grown on rice. And so we make this really delicious syrup, and the bees love it. And then we did these experiments at Washington State University, as you saw, and we fed the bees the extracts in sugar water. All beekeepers practically use 50% sugar, 50% water to supplement and feed their bees, especially commercial growers. So we did this. We fed them the extracts. I just need to speed this up. And an example of a commercial bee operation where the sugar water is being fed to the bees. And so we just introduced the extracts into the bees. And so the conclusion of screening about 20 different polypore mushrooms, very interesting. The red-belted polypore, chaga, a birch polypore, amadou, which my hat is made of, and the red reishi gave us extraordinarily interesting results. Now, Dr. Steve Shepard, as an entomologist at 39 years studying bees, I'm unaware of any materials that extend the life of worker bees more than this. This is the time the bees are foraging the most. And what's happened now is bees typically live for about 30 days, 9 days foraging. And when you see bees on a flower, it's the last few days of their life. That 9-day foraging period has been cut to 4 days in the past 5 to 10 years. So the bees are prematurely dying. The nurse bees abandon the brood, not taking care of the babies. They become worker bees, they fly away. It's a slippery slope downhill. So the fact that we nearly doubled the longevity of bees, high significance here, with the amadou mushroom is extraordinary. So we've increased the ability of the bees to forage, and we increased longevity with the extracts. Then when the red-belted polypore, which the bears introduced, we found at the right concentration, it also had a doubling effect on the longevity of bees. And then with the bee viruses, with chaga, which if you notice in the BioShield Biodefense Program, reduced flu viruses, it also, on a dose-dependent basis, the controls, the viruses go up, and the increased concentration, the viruses go down in the bees. And then with the red reishi, also in the BioShield program, reduces flu viruses, H3N2, H1N1, H5N7. It also, the controls go up. On a dose-dependent basis, the viruses go down. So we increase the longevity of bees by doubling them, and we're reducing their viruses by more than 90%. So we thought. So here is a summary of our initial first year of research. Tremendous increase in viruses, tremendous decrease in viruses when given these 4 different species. Okay, so the deformed wing virus is identified now as the #1 virus that's harming bees. It is a pandemic that has spread from Asia all over the world. There are no, I'll come to this. So in 2016, 5 peer-reviewed articles identified honey losses being primarily caused by the deformed wing virus. Now there's the Lake Sinai virus, the Israeli apiary virus, and the black cell queen virus. These are also viruses that harm bees. But the deformed wing virus is the primary virus of concern being injected by the varroa mites into the bees. So here is an extraordinary statement, is that 100% of honeybee colonies in the world are now infected there is a global pandemic, the bees, they wander and they go to other hives. And so now all wild bees are infected with the deformed wing virus that have come from commercial honeybee operations. And so Dr. Jay Evans, who's the renowned virologist at USDA, I talked to him about 2 weeks ago, and he said that he has a researcher from England that came over to try to get a virus-free strain of bees. And he said, Paul, I haven't seen a virus-free strain of bees in over 10 years. So the native bees are being decimated right now. They're going extinct faster than we can determine. I talked to one entomologist. I go, how do you know if they're extinct? And he goes, Paul, if you haven't found them in 10 years, does that mean that they're extinct? You know, it's like a rhetorical question. So this is a huge, huge problem. And so here is our latest results, folks. We hit it out of the park. Amadou and reishi against the deformed wing virus, now with a new preparation, reduces the virus more than 1,000 to 1 in a week to 2 weeks. Against the Lake Sinai virus, this is outdoors, this is with queens, and we've now more than 10,001 of the Lake Sinai virus. So we have been able to potentize this, and now we're doing 1 drop per 1,000 drops of water. This stuff is so incredibly potent. So, and then against the black queen cell virus, all queens now, unfortunately, have this virus as hurting the queen stocks that, you know, are distributed to beekeepers. Also against the black queen cell virus, we're seeing with reishi and chaga more than a 5 to 100 to 1 reduction in these viruses. So I'm very thankful and happy that I did receive a patent on this October 14th. And they now, and my true intention is, if I can afford it, I want to open source this technology for everybody in the world. So, I mean, think of this. They use the biggest search engines available in Chinese, Japanese, Russian, English, Spanish, you know, all languages. And I got the report back, and it said, no prior art, except, Paul, your article in Harrismith magazine, you mentioned the bees going to your garden giant patch. And at first, you know, I have to say, my ego went, whoa, you know. And then I got, really? We all grew up with Winnie the Pooh. We all knew that bees were attracted to rotted logs, and I'm the first one to discover that bees are attracted to mycelium because of immunological benefit. I gave a talk into a bee conference with 900 bee scientists, and I milked it. I said, stand up now if you've ever heard of this. Where did you see it? Where was it published? Dead silence. So the international patents have been issued. I have to name something after myself. The Stamets mushroom bee hypothesis, and it has a synergistic effect in being helping the bees, and that is mycofactor number 6. Yes. I have three minutes left, so okay, this is crazy. We go back to p-cumaric acid. I get a hold of the NIH virology. I said, you sampled over 2,000 samples. I got 77 strains of agarikon. We had 3 that were active. I want to test 77. They wrote me back immediately, said, we not funded by the BioShield program. We won't do bio-guided fractionation for you. You have to send in a pure structure. We're happy to work with you, but the structure you send in, a pure molecule, has to be juried that no one ever discovered it before. And cytologically, does a virologist think it will make sense? So I'm going, oh, they will test more, but I need a molecule. I don't have millions of dollars. I can't do bio-guided fractionation like I did with Scott Franzblau and the University of Mississippi. What am I going to do? And I remembered p-cumaric acid. So then, rather than bio-guided fractionation, Louis Pasteur has a great quote, chance favors the prepared mind. So I did the best of all scientific methods, I guessed. I submitted 20 molecules, 10 of them highly active, more potent than antiviral drug controls, all surrounding p-cumaric acid, which comes from wood, from the mycelium breaking it down. 5 of these molecules are highly active against HPV. And I'm sorry to tell you this, but most of you under the age of 30, 90% of you are carrying an oncovirus called the human papillomavirus, which is a ticking time bomb that causes cervical cancer in women and other types of cancer as well. There is no good antiviral medicine against HPV, 5 molecules that I discovered, highly active against HPV. I did it in 5 minutes by guessing. Okay, so not on my birthday. But so again, flu viruses, herpes, norovirus, hepatitis, pox, and also we have polio. And so that is mycofactor number 7. So I want to just introduce to you, there is a mycofactorial equation that I have designed here. There are the different factors in my life that have been strung together. And now we know we can limit disease transmission by insects to plants. Think of that. I think we have the gateway now of mitigating all viral vectors that harm plants, by controlling the diseases in the insects prior to transmission. So what's next? What's next in this mycofactorial equation? I leave that to you. Many of you, these ideas will synergize, and you might have epiphanies, happening all over the audience right now. So we are be-mushroomed. I believe that scientists across disciplines need to work together. Mycodiversity is our biosecurity. And I want to mention that mushrooms, trees, bears, birds, and the bees, we all have evolved to be within the matrix of mycelium. And think of this, for 10,000 years ago, we started agriculture. What did we do? We deforested. That began to dismantle the immunological networks of nature. I think I found something fundamental to the foundation of nature, that the mycelial networks control the diseases of the inhabitants that exist within those ecosystems. This is truly paradigm shifting. That goes way beyond human health and bees are the second most well-studied animal in the world. So I can't believe that I discovered this. I'm not really a beekeeper. I'm a mycologist, And for me to discover this when all these other great scientists have been working so hard for literally hundreds of years cumulatively is truly amazing. So I want to end with just a 2-minute, I'm going to be 2 minutes over, 3 minutes over, a 2-minute short movie Louis Schwartzberg and I are putting together. And I want to just suggest to you, let's celebrate decomposition. Let's let it rot. Mycodiversity is biosecurity, and Bill, with your tolerance here, here's a 2-minute movie. But I have constructed something new called the Mycoverse. It's my multi-generational platform for helping people for hundreds of years into the future. So stay tuned on the Mycoverse, because dark matter, the neurons, the mycelium, we all conform and share the same archetype of networking. And so this should be the movie.
Mushroom mycelium represents rebirth, rejuvenation, regeneration. Fungi generate soil that gives life. The task that we face today is to understand the language of nature. My mission is to discover the language of nature of the fungal networks that communicate with the ecosystem. And I believe nature is intelligent. The fact that we lack the language skills to communicate with nature does not impugn the concept that nature is intelligent. It speaks to our inadequacy for communication. If we don't get our act together and come in commonality and understanding with the organisms that sustain us today, not only will we destroy those organisms, but we will destroy ourselves. We need to have a paradigm shift in our consciousness. What will it take to achieve that? If I die trying, but I'm inadequate to the task, to make a course change in the evolution of life on this planet, okay, I tried. The fact is, I tried. How many people are not trying? If you knew that every breath you took could save hundreds of lives into the future, had you walked down this path of knowledge, wouldn't you run down that path of knowledge as fast as you could? I believe nature is a force of good. Good is not only a concept, it is a spirit. And so hopefully the spirit of goodness will survive.
Thank you very much, folks.